So, today's video is on tissue fluid. So first we're going to look at the lymphatic system because this is an important part of our presentation today. So the lymphatic system is part of our circulatory system, so this is the transport of blood around the body. And the lymphatic system is an open system, so this means that the fluid in the lymphatic system is not confined to the vessels for the entire time. So it means that they're open-ended, they can let the fluid out of their vessels. And the lymphatic system is a network of lymphatic vessels. So now we're going to look at blood versus tissue fluid versus lymph, because these are the three fluids that are going to be important in the process we're going to learn about. So firstly, blood. It's the fluid that circulates blood vessels, so this is pretty self-explanatory. Tissue fluid, or interstitial fluid, is the solution that bathes cells in multicellular organisms. So this is the fluid that surrounds all of the cells in our body. And lymph circulates the lymphatic system that we've just learnt about. So more on blood versus tissue fluid versus our lymph is that blood contains water, glucose, amino acids, fatty acids, carbon dioxide, oxygen, protein and hormones, along with many other types of cells, but these are the molecule components in the blood. This is the fluid that it contains and it transports. Tissue fluid contains the same as blood, but not the larger molecules, and this is because the larger molecules will not be able to leave the blood vessels. And then we have lymph, and lymph contains exactly the same as tissue fluid. So you can see that these three fluids, even though they have different names, are mainly the same composition of molecules. So we can see that these three fluids, although named different things, are the same fluid just in different parts of the body. So first we're going to look at the journey of the fluid. So first our fluid starts as in the capillaries as blood. And then out of these capillaries the fluid is forced out into the tissues where it becomes tissue fluid. So it was blood, it's forced out of the capillaries and becomes tissue fluid when it reaches into the tissues. And then our tissue fluid either returns back to the capillaries or it goes into the lymphatic system. So every day 20 litres of fluid seep out of our blood vessels. So this is this stage here, 20 litres of our tissue fluid in the blood seeps out of the capillaries into the tissues. And 17 litres of these 20 are reabsorbed back into the capillaries afterwards. And this is where it's taken back to the heart. So that leaves 3 litres that are left in the tissues. And these have to be returned to the heart via the lymphatic system. So this is our network of lymphatic vessels. So how does this fluid exchange take place at the capillaries? So fluid exchange is determined by two factors, and this fluid exchange is the tissue fluid in the blood being forced out into the tissues. So it is determined by hydrostatic pressure, and this is the pressure caused by the pumping of the heart. And it's also determined by the water potential of the blood. And we'll be looking into these now. So hydrostatic pressure at the arterial end. An arterial end means it's the end where the arteries and the arterioles lead onto the capillary. So this is where the blood is coming from the heart. So we have to know that blood moves from a high to low pressure. So it moves down a pressure gradient. So this hydrostatic pressure caused by the pumping of the heart causes a higher pressure inside the capillaries than the tissues. So that makes sense. So if our blood is flowing through our capillaries here, it has the pressure, the hydrostatic pressure caused by the pumping of the heart. It has a higher pressure than our tissues. And this causes our fluid to move into the tissues from the blood, because our blood moves and our fluid moves from high to low pressure. So it will move down this pressure gradient into the tissues. But we also have to take notice of ultrafiltration. So this means that only small molecules are forced out under pressure, because the large molecules cannot leave this capillary wall. And that is why our tissue fluid and our lymph don't have the larger molecule components as our blood does. So what opposes this hydrostatic pressure? Because our hydrostatic pressure does let the tissue fluid be forced into the tissues, but there must be some forces opposing this. So the hydrostatic pressure of the tissue fluid outside the capillaries resists outward movement. So this is the hydrostatic pressure of the tissue fluid already in the tissues, and this resists the pressure of the tissue fluid inside the blood moving into it. However, because our pressure in the capillary is larger than this hydrostatic pressure already in the tissues, our tissue fluid can still move out of the capillaries. And also water potential. Water potential opposes this hydrostatic pressure, but we will be looking at that in a moment. So hydrostatic pressure at the venule end, so this is where it leads on to veins to be taken back to the heart. So as the tissue fluid has all been forced out of the blood capillary, the pressure inside the capillary is now lower. So as all of our tissue fluid has been forced out into the tissues, eventually the pressure inside our capillaries will drop below the pressure of our tissues. So this, we have to remember, blood moves from a high to low pressure. So if our pressure is now higher in our tissues, the tissue fluid will now be reabsorbed back into the capillary where there is a lower pressure. So now we're going to be looking at water potential. So as the tissue fluid is forced out due to hydrostatic pressure at the arterial end, the water potential in our capillary lowers. So as we go down this capillary and all of the tissue fluid is moving out due to our hydrostatic pressure, our water potential is lowering as we go down. So via osmosis, water moves back into the capillary where there is a lower water potential, because we know osmosis is the movement of water from a high to low water potential. So at the venule end, where our water potential is lowest, osmosis will move our water back into our capillaries. So that's how the water gets reabsorbed back into the capillary at the venule end, because it has a lower water potential than the tissues surrounding it. So how does our tissue fluid return to the heart? Well, as we know, some of our tissue fluid isn't reabsorbed. So the tissue fluid that is reabsorbed into the capillaries will be taken back to the heart via the veins. But our tissue fluid that remains, what happens to it? So our excess tissue fluid that's left in the tissues and is not reabsorbed is absorbed by our lymph vessels in our lymphatic system. And these lymph vessels transport our tissue fluid to the subclavian vein, so that's next to the heart. And this drains the contents back into the blood system. So our tissue fluid is taken up by these lymph vessels, transported to the subclavian vein, where it drains the contents back into the blood system to be re-pumped by the heart. So how does this happen? Because we know that in the lymphatic system, our tissue fluid has already lost a lot of pressure from when it originally started from the heart. Well, the tissue fluid uses some of its remaining hydrostatic pressure to force itself through the lymphatic system, but the main part is the contraction of body muscles, and these contractions squeeze the lymph vessels, forcing the tissue fluid up the lymph vessels towards the heart. And that means the lymphatic system has to have valves, and these valves keep the tissue fluid flowing in the correct direction up to the heart. So what are the other uses for our lymphatic system other than draining excess tissue fluid and transporting it back to the heart? Well, the villi in our ileum have lacteals, and lacteals are little lymphatic vessels. As you can see here, this is our villi, and it has a lacteal in the middle. And these lacteals take up large molecules that cannot diffuse into the blood. For example, chylomicrons. So these chylomicrons are uptaken by these lacteals. And these deposit these molecules into the bloodstream at the subclavian vein. So these molecules that cannot be diffused into the blood are transported into the bloodstream by being taken up by these lacteals and transported via the lymphatic system. So this means that when our heart pumps, these molecules are taken to the rest of the body, when they would usually not be able to diffuse into the blood.